Let's look at Chapter 3. Chapter 3 is about protein structure and function. Remember the chemical evolution, that there were once small, very small molecules, and gradually through time they became larger and larger and more ordered. They became ordered because they have more energy and they're more stable. And we know that the energy is located in the bonds. So there's four important macromolecules. There are proteins, fats, nucleic acids, and sugars. So proteins will be one of the first macromolecules that we talk about. It's one of four. In this chapter, we'll talk about the building blocks of proteins. There are 20 amino acids, and these amino acids are the building blocks of proteins. What makes each amino acid unique? It's their side chain. Then once we talk about the content of what makes a protein, we'll talk about structure. There's four levels of structure: primary, secondary, tertiary, and quaternary. Then after that, we'll talk about protein function and the diverse roles that proteins serve in living cells. Cells produce tens of thousands of distinct proteins. In fact, we're made mainly of proteins. When I say that you look mighty nice today, actually, am I not talking about your proteins? They're vital and they're tremendously versatile. So we cannot live without proteins. The building blocks of proteins are amino acids. In fact, they have found amino acids on meteorites. So they've been able to survive under space-like conditions. So the question is were proteins what sparked life on Earth? Were proteins necessary for the first cell? As I mentioned before, the building blocks of proteins are amino acids, and in fact there's 20. Some books say that there are 21, but what makes each amino acid unique is the side chain. So what does an amino acid look like? It says it's composed of a central carbon, and it's bonded to a hydrogen, an amino group, which is NH2, a carboxyl group, which is COOH, and then that side chain, which we call the R group, and that's what makes each of the 20 amino acids unique. So here's a diagram of a non-ionized amino acid. In other words, if it's non-ionized, there's been no transfer of electrons. And you see on one end there's the amino group, on the other end is the carboxyl group, and in the center is the carbon. Up above is a hydrogen, and down below is the side chain. You're going to need to know this. I'm going to give you 5 points extra credit for actually knowing how to draw this diagram. It says here in water, amino acids and carboxyl groups ionize to ammonia and COO. So what does ionization mean? And I know that you remember when an atom is ionized, it has either transferred an electron, so one atom is lost an electron and another one is gained one. And you can see in this diagram that the carboxyl group actually lost its proton and the amino group gained a proton. So the amino group acts as a base because it attracts a proton, and the carboxyl group on the other side of the amino acid acts as an acid because it donated its proton. Keep in mind, when we talk about a proton, are we not talking about hydrogen? Remember, hydrogen has one proton in the nucleus and no neutrons. So what happens when there's been a transfer of a proton? Well, that means that the amino acid becomes charged. One side is positive and the other side is negative. Well, water is charged also, so that makes an ionized amino acid very reactive in solution. Here's the ionized form, and you can now see that the amino group has a proton and it has a positive charge. On the other side, the carboxyl group has lost a proton or a hydrogen, and it now has a negative charge. So we see that it's ionized and it's reactive in water. Okay, going back to what makes every single amino acid unique is the R group or the side chain. So if you look in your book, you'll see the 20 amino acids and how they differ truly is by this side chain known as the R group. So the side chain can be charged, uncharged, and polar or nonpolar. In figure 3.2, you see all 20 amino acids. So the truth is do you have to know this for the test? No, you don't have to know each of these, and you don't have to know what the side chain looks like, but you do have to know that it's the side chain that makes every single amino acid unique. So remember, if it's charged and polar, it's hydrophilic. And remember, hydrophilic means water-loving. Isn't it true that water's charged? And if the amino acid's polar, then it too must be charged. Birds of a feather flock together. Water interacts with charged molecules. On the other hand, there's also nonpolar amino acids, and these are hydrophobic. Remember, hydrophobic means water hating, and therefore it does not interact with water. Well, there's a way that you can put these into groups, but I'm going to be honest with you, class. You saw them in the previous diagram, but you do not have to know this for the exam. So several questions you can ask about the R group is, does a chain have a negative charge? Does it have a positive charge? Is the side chain uncharged but have an oxygen atom, or is it just really totally nonpolar? So these are how it's categorized and put into the groups that you saw in the previous slide. Okay, so as I mentioned before, proteins are the first of 4 macromolecules that we'll be studying. So another name for macromolecule is polymer. But let's take a look at the word. Macro means large, and you know what a molecule is. A molecule are a bunch of atoms bound together. So this means a very large molecule. What does polymer mean? Poly means many. So poly means many units put together. So keep in mind the building block of a protein is an amino acid. It is also, we can say it's a monomer. So another name for building block is monomer, mono meaning one. So these one units get together to make many, or polymers. Amino acids are the monomers that make up proteins. I love this diagram. At first glance it looks so simple, but the truth is how do we get these large macromolecules? How do we get these large polymers? Well, it looks like we're binding together these basic units. In the case of proteins, we're putting one amino acid together with another, and then we get this very large chain, and before you know it, we have a macromolecule. Well, how do we form bonds between these building blocks or monomers? And the way this is done is through condensation or dehydration reactions. And this bond is the result of the loss of a water molecule. You know what I always think about when I think of dehydration? I always think about a hot summer day in El Paso and about wanting to eat a slice of pizza and a cold beer from either a can or from a bottle. It doesn't matter. And when you're holding that long neck beer, you see those little droplets of water condensing on the glass or condensing on the can. There's a loss of water. You can see the water dripping down. Condensation reaction. The opposite of that is hydrolysis. In my head, you know what I think of when I see the word hydrolysis? I always say in my mind hydrolysis, because hydro means water and lysis means break. Hydrolysis is the opposite of a condensation reaction, and water goes in to break the bond between the monomers. I love this figure. Again, at first glance, it looks sort of simple, but really it really is quite complicated. So the first part, A, is a condensation reaction. Remember, condensation is like those little water droplets on a can of cold beer in the summertime. Water goes out. Water goes out. And notice here, a bond is formed between one monomer and another. You see H and OH make water, and it comes out, and the bond is formed. The opposite is hydrolysis. Water goes in, and notice that it breaks the bond. Polymerization requires energy. And you know, that sort of makes sense. Isn't it true that potential energy is stored in the bonds? So the larger the molecule, the more bonds it has. So if it has more bonds and each of those bonds represent energy, then it takes a lot of energy to build a large molecule. Okay, so monomers can't just self-assemble, it requires energy. And keep in mind, when you're building something, it's sort of the opposite of entropy. And you guys remember what entropy is. It's disorder. So small molecules represent disorder and large molecules represent order. And it takes a lot of energy to build a large molecule. So it says here, during the chemical evolution, polymerization would only occur if the amino acid concentration was very high. So they had to be present for actually the reactions to occur, and that makes sense. So what is the special bond that forms between one amino acid and another? This special bond is called the peptide bond. I promise you it's going to be on the exam. And peptide bonds are always formed between one carbon and a nitrogen because they line up like this. The backbone is NCC, 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 NCC. So the carbon of one amino acid binds with the nitrogen with the next one, and this is the peptide bond. It says it's very stable because a pair of valence electrons on nitrogen are partially charged and share the carbon-nitrogen bond. So it says here the peptide bonds have characteristics of a double bond. Remember, single bond is 2 electrons and a double bond has 4 electrons. This diagram shows how amino acids line up. NCC, NCC, that's the backbone, correct? Can you see it? And notice that the carbon of one amino acid is going to bind with the nitrogen of the next amino acid. And it's going to be through condensation reaction. You can see highlighted in blue is the oxygen and 2 hydrogens. When they go out, then a peptide bond is formed between carbon and nitrogen. In addition to NCC, NCC, isn't it true that the peptide bond is forming the backbone of a protein? It's truly NCC peptide bond, NCC peptide bond, NCC peptide bond. So it says here, when that happens, then other things begin to happen too. There's a chain reaction. Once all of the amino acids start to bind, then the R group orientation on the side chains extend out and they start to interact with one another. The other thing that we notice is directionality. There's the nitrogen side, which is called the N-terminus, and then there's the carbon side, which is called the carbon terminus. And then there's flexibility. Instead of it just being linear, actually it's a very, very, very flexible backbone. I love this diagram. It shows NCC, 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 and in between each of the amino acids is a peptide bond. Then you see all the side chains, and they're going to start to interact with one another. And so one side is the nitrogen terminus, and on the other side is the carbon terminus in the carboxyl group. This diagram shows that as soon as they start linking together through peptide bonds, now a protein is very flexible. It's not just two-dimensional. Isn't it true that once it starts to bend and fold, it has a three-dimensional shape, just like we do? Notice again, the nitrogen side is from the amino group, and at the other end is the carbon from the carboxyl group. There's the nitrogen terminus and the carbon terminus. Now, you're going to read in your book sometimes that they'll call a protein a peptide. I've never heard of anyone really talk about an oligopeptide. Now, I have heard polypeptide, and all of those are used synonymously with protein. So if you hear the word peptide, it means protein. If you hear polypeptide, it also refers to protein. But keep in mind, by definition, a protein has to have more than 50 amino acids. It truly is a macromolecule. So what do proteins look like? If we're made mainly of proteins, then you can imagine there is a lot of diversity of size and shape and chemical properties. So form and function go hand in hand. So proteins serve diverse functions, so therefore they must have also diverse shapes also. So here are some examples. Collagen is up at the top, and you know that collagen is found in creams and in bone broth, and it's to make your joints nice and strong. Now that I'm an old lady, I think about it. We'll talk about the Tata box later on for DNA replication. A porin is found on the cell membrane, and it allows water to go in and out. And then there's chymotrypsin, and this one looks like a solid ball, like with hair coming out. Okay, so there's basically 4 structures to proteins, and you'll have to know this for the exam too. Primary, secondary, tertiary, and quaternary. First level, second, third, and fourth levels. So what is a primary structure? What is its first structure? And this is what makes a protein unique. It's the sequence of amino acids. So it's the number of primary structures is practically limitless, and so it's just like beading a necklace. You could have a whole bunch of beads, but you can put them in different orders and put the shapes in different ways, and you could make a limitless number of different necklaces. The same thing applies for amino acids. The other way I look at it too, it's like amino acids are like letters of the alphabet. We can arrange the letters to make small words and big words, and we can use those words to shape sentences. And there's a limitless number of sentences that we can make. You know what? The primary structure is the most important structure of all the structures, and I promise you, this will also be a test question. So it says here the primary structure is fundamental to the higher levels of protein structure. In other words, it's just like I said before. Out of all the levels of structure, which one is the most important? It's the primary structure. So the secondary, tertiary, and quaternary level are all based on the sequence of amino acids. It says here a single amino acid change can radically alter protein function. The classic example is sickle cell anemia. So in the upper part of the slide, you see a normal red blood cell, and this is associated with glucine, or the 6th amino acid in a chain. But notice there's a mutation in part B. Instead of glucine, you see VAL for valine. And notice what it's done to the red blood cell. Instead of looking like a frisbee, now it looks like a sickle. And because of the sickle shape, it's not able to hold enough oxygen. Okay, the secondary structure is formed by hydrogen bonds. You know, when I think of the secondary structure, I think of hairdos. Yeah, you got me right. I said hairdos. I think of Shirley Temple, and I think of Bart Simpson. Shirley Temple had ringlets, and Bart Simpson's hair goes up and down. And that's what the secondary structure is for actually a protein. So there's two secondary structures, alpha helices or beta pleated sheets. Alpha helices are the ringlets like Shirley Temple hair, and beta pleated sheets, that's Bart Simpson's hair. So in this diagram, you can see the two types of structures, secondary structures. And notice that they're held by hydrogen bonds. Do you remember what a hydrogen bond is? It's weak opposite electrical charges attracting each other. And of course, hydrogen is always involved. So this is what maintains the ringlet structure or the pleated shape. The tertiary structure. The tertiary structure of a polypeptide results from interactions between R groups. So basically what's happening here is that other types of bonds are being formed besides hydrogen bonds. And so now you see that there's really this complex three-dimensional shape that is forming, and they're held together by different types of bonds. So it says here the contacts cause the backbone to bend and fold, and this bending and folding is distinctive to each of the proteins, and it creates this three-dimensional shape. Okay, so what are the 5 ways that the R groups interact to form these like permanent interactions and bending of the protein? I promise you, you also need to know this for the exam too. So in addition to hydrogen bonds, which are opposite partial charges or weak opposite charges attracting each other and maintaining the shape, there's also hydrophobic interactions. Remember hydrophobic? Hydrophobic, water hating. So water forces hydrophobic side chains and pushes them together. There's also van der Waals. These are weak electrical interactions and they occur between hydrophobic side chains. There's also disulfide bonds. They form bridges and sulfurs on both sides of this sulfur bridge. And then ionic bonds. Well, you've heard of ionic bonds before. Isn't it the transfer of electrons from one atom or molecule to another? And this forms an attraction between opposite charges. This diagram shows all of the interactions: hydrogen bonds, hydrophobic interactions, van der Waals, disulfide bond, and ionic bond. This is a wonderful diagram because it shows exactly what they mean, and you'll need to know that for the exam. In part B, it shows you how you can have alpha and beta pleated sheets. In the first diagram, you only see alpha helices, and then the second one you begin to see a combination of both alpha and beta structures, and in the last one you see these little yellow hooks, and these represent disulfide bridges. Okay, the quaternary structure. The quaternary structure is the 4th level. So what is it? It's putting a lot of polypeptides or protein units together to make like a lean machine, and this makes one single structure. So it says here it's the binding of these polypeptide units to make the last level, and in some ways they are considered molecular machines. One of the most famous one of all time is hemoglobin. If you take a look at the diagram and on the right hand side, you'll see that it says alpha 1, alpha 2, beta 1, and beta 2. So you see that there's 4 subunits. There's 4 polypeptides. They've come together to do what? Their function is to bring oxygen to your cells via the red blood cells. In A, we see the CRO protein, which is a dimer, di meaning 2. So you see that they both almost look like mirror images of each other. So folding and function goes hand in hand. And it says here protein structure is hierarchical. Well, what does that mean? It means primary, then secondary, then tertiary, then quaternary. But all of these are based on what structure? The primary structure. And what does the primary structure represent? It's the sequence of amino acids. So all of these levels of structure allow for amazing diversity in protein form and function. I love this summary table in 3.1. It shows you all the different levels, it gives a description, and it shows how they're held together, and it even gives you an example. If you can look at this table and understand what's going on, I realize that you have understood what I've been talking about. Normal folding is crucial to the function of a protein. So oftentimes the folding of a protein is very spontaneous, and it's because of hydrogen bonds and van der Waals interactions. But sometimes proteins can become denatured. So what do we mean by denatured? It means that they can become unfolded. You know when someone says, I'm so anxious or nervous, I feel like I'm unraveling? Well, isn't that what that means? That you're unfolding a protein. And if it's unfolded, then it won't be able to function. So form and function go hand in hand. So oftentimes to keep a protein folded properly, we need other proteins. And these proteins are called molecular chaperones. And they help to fold proteins correctly. I sort of think like when I pull clothes out of the dryer and I have to fold them properly before I put them into drawers, that there's no way that they're going to fit nice and orderly unless they're folded properly. So here's an example of a protein. On the left-hand side, you see that it's a three-dimensional shape held together by hydrogen bonds, disulfide bridges, but when it becomes denatured, it literally unravels. And when it unravels, it has broken the hydrogen bonds and the disulfide bridges, and it is no longer a functioning protein. It is denatured. Protein shape is flexible. Each protein has a characteristic folded shape that is necessary for its function. So in some cases, if the shape is disordered, then it's inactive. For a protein to become active, then it folds and it has this active conformation and then begins to work. So we can say that some proteins are regulated by controlling when or where they are folded into active shapes. Now, there's sometimes that proteins are misfolded, and this misfolding can be dangerous and even be considered infectious. So these misfolded proteins are called prions. And it says the amino acid sequence does not differ from a normal protein, but the shape is radically different. Now these prions can induce normal protein molecules to change their shape and alter their form. In this example here, you see on the left-hand side a normal protein and then an infectious one. So the amino acid content is the same, but the shape has changed. Protein functions are as diverse as protein structures. So why have we been studying proteins? Because they're crucial to most tasks required by cells. So what are these tasks? Catalysis, which speeds up chemical reactions. Defense, that's antibodies used to attack pathogens. Movement, proteins are used to move cells or molecules within cells. Signaling conveys signals between cells at the membrane. Structure, it helps with shaping the cells and comprise body structures. And lastly, transport. It allows molecules to enter and exit cells or carries them throughout the body. Of all of these functions, oftentimes catalysis has been considered the most important protein function. Well, there's a subset of proteins that carry out catalysis, and these are called enzymes. So an enzyme is a protein that serves as a catalyst, and it acts on substrates at the active site. So substrates are reactants, and they fit inside an enzyme in the active site. Enzymes bring substrates together in a precise orientation, and that makes the reaction more likely to occur. Isn't this another way of saying that it speeds up the reaction? One enzyme is needed for one specific reaction. It's akin to a lock and key model, where the lock is the active site and the key are the specific substrates that fit in the active site. Here's an example of a huge macromolecule, and within you see red and blue molecules representing the substrate, and only those molecules can fit in the active site for the reaction to occur.